So as chair of the Federal Committee of the Plant Conservation Alliance, I am pleased to introduce Mr. Wesley Knapp. He is the chief botanist at NatureServe, a leading biodiversity conservation nonprofit in the United States. Wes has over 20 years of experience working in the NatureServe network as a botanist and ecologist with both the Maryland and North Carolina Natural Heritage programs. And he has extensive field experience across much of the United States, along with forays into Australia, Canada, and Central America. His research ranges from plant extinction to plant discoveries. And among his publications is the 2021 Vascular Plants of Maryland, USA, a comprehensive account of the state's botanical diversity, which is freely available through the Smithsonian Scholarly Press website. Without further ado, speaking to us about identifying and preventing plant extinction events, let's welcome Mr. Wesley Knapp. I give you the floor.
All right, so first off, thank you all for having me today. I love talking about rare plants and conservation. And I'm going to touch on that 2021 paper on plant extinction events, 2020 paper on plant extinction events, and then branch off from there on some new work that we're doing to prevent plant extinction events. As just mentioned, a lot of this really stemmed from this paper in conservation biology that I was honored to lead among some of the who's who in American botany, really. Like Anne Francis is on the call. She was a wonderful partner on this. I think I saw Bonnie Heidel's name. So a lot of member program botanists as well in the Natural Heritage Network, kind of the NatureServe network. This is open access. Feel free to download it if you're interested after the talk. But I'm glad my roots as a heritage botanist were brought up because that's really been formative for my career. It's been formative for my passion to identify and prevent plant extinction events. And I remember getting my dream job as a botanist with the Maryland Natural Heritage Program way back in 2001. So I'm dating myself. And I was really unequipped to do the job of go out and find rare plants and work to protect them. So I studied the rare plants of Maryland a lot, and I'll keep this short, but I was really enamored by this plant here, Nuttall's micranthemum, micranthomoides, because it's the only component of the flora that has a rank of GH, SH, and X, so it's extinct. And I didn't know we had extinct plants in our backyards like that. To think that we had extinct plants in Maryland wasn't something I was accustomed to thinking about. So I studied as much about the species as I could, attempted to rediscover it. Of course, I couldn't because it's extinct. But this is really the opposite of charismatic megafauna. It's a cryptic intermudflat species, last observed on the 13th of September 1941. This is one of the duplicates. And then Sumeria, never seen again. We have no idea really why this went extinct, which is one of the conundrums of extinct plants we'll talk about later. But it also led me to realize as a community, we don't appreciate our extinct plants. I'd go to conferences and talk to botanists and ask what the extinct plants were in their states, and I'd usually get blank glares, or people were aware of Franklinia altamaha, Franklinia tree. I'm sure most of us have heard of this. This is a fascinating story of a plant now that is extinct in the wild, only known from the gardens, collected by Bartram. And he documents it in the book Bartram Travels. I actually have a copy behind me, and it's pretty uncelebratory. It just mentions how he's going down the trail, finds this amazing shrub, he's going to name it after Benjamin Franklin. Decided to introduce it into the horticultural trade, and thankfully he did, because this is among the last times this plant had ever been seen by man. So we realized we have a poor understanding of our extinct plants, so we wanted to learn what we could. So we wanted to compile a comprehensive list of the plant taxa believed to be extinct in the United States and Canada, the continental United States and Canada. We wanted to calculate an extinction rate as a baseline for the Anthropocene. So here all the time in the literature that extinction rates are expected to increase as we move through the Anthropocene, but we don't have a good understanding now of what our extinction rates are. We wanted to draw attention to these extinct plants to drive future conservation efforts like inventory and taxonomic study. And then frankly, an extinction event is a conservation failure. So we wanted to learn what we could from these failures to prevent future extinction events. Now, I want to take just a quick second to mention that extinction is a hypothesis, and I hope we're wrong about every one of these plants. I hope they're out there lurking somewhere. And we have two categories at NatureServe. We use presumed extinct, GH, or presumed extinct, which is X, extinct, and globally historical, which is H, which gives some hope of a rediscovery. So I'm putting both of those categories together here for today for simplicity. We're going to mention that there's no confirmed sightings for over 25 years. We're looking only at extinction since European settlement, so we're not looking at, a Pleistocene or late Pleistocene Triassic extinctions or anything like that. And we're not talking about extirpations or localized extinction events. We're looking at global extinction events where plants are no longer known to exist. Now, the study area is frankly artificial. It's the continuous United States and Canada, but it parallels the Flora of North America series well, which is unfortunately not finished, but a good product nonetheless for comparative analysis on the flora. We didn't look at Central America. Maybe someday we can do that. Or we did not look at Hawaii and Puerto Rico because of issues of island biogeography, but that would be wonderful to include into the total data set. And we didn't include bryophytes or lichens, and there is one extinct bryophyte from Canada, this neomaculated nitida, which could frankly be another area of study to look at bryophytes. This is a real study on vascular plants. And my computer, here we go. So the methods are what you'd expect at the preliminary stages, which is a lot of literature review, primary monographs, literature, floristic studies. And I owe a great debt to the Smithsonian Library for letting me just hunker down for a few days and go through all those manuals I don't have access to. We also did a lot of conversations with botanists across the United States and Canada, and that includes those NatureServe member programs and the data housed in the NatureServe database, where we look for GH and GX plants. And if you're not familiar with NatureServe, we're a network of member programs that stretches across the United States and Canada. So our member programs are shown there in blue, and then in green shows kind of the footprint of projects we've worked on across the globe. And we have this standard ranking methodology with 5 being the most common and 1 being the most rare. But we have these 2 ranks, GH and GX, that we do not use very often, thankfully, because they mean extinct, which we could start to build upon this kind of a structure of what plants were extinct in the US and Canada. But as you can imagine, not all names are created equal. We had to come up with a process on how to vet all the names that are found in the literature and at NatureServe to make sure that we're comparing apples to apples and meritorious entities with one another. Because if you just pull data from a database, you're going to end up with hundreds of names. Like we had 150 names at first, but I mentioned not all these names are created equal. We have plants like Franklinia, which is universally accepted as a distinct species. Sometimes it's put into a different genus, but it's always recognized as a distinct species. But then you have these other rather dubious names like Rubus fryi, which is either extinct and was endemic to West Virginia, or it's not real. And the stakes are really high when you have taxonomic disagreement at that scale. So we came up with a novel method we call the Index of Taxonomic Uncertainty, which is basically the scientific consensus of a name, to make sure all of our names are being compared equally. Now it's a little tricky. We wanted to look at treatments with different authorships, so the floras and manuals of different authorships, because it would be a bit unfair to look at all the treatments I've written for Juncus and say everyone agrees in Juncus about the name Juncus longii, which isn't always true. You want to look at different manuals and treatments, come up with the true scientific consensus. And we excluded databases like USDA Plants or NatureServe or BONAP because we wanted to go right to those experts who are working to write treatments to delineate species. And we used a standard school grading system for this, A to F, where A is a universally accepted entity like Franklinia altamaha. B is something with majority acceptance like Juncus purvitus, which was known from one place in Massachusetts. It is most often recognized as a distinct species, but occasionally Gleason and Cronquist, namely, said this was a waif of a different species from Europe. We have plants that are minor taxonomic acceptance like Narthecia montanum, where more often than not, they're not recognized as distinct, but still numerous current sources argue that it is a distinct species or a distinct taxonomic unit. And then we have groups like D and F, which are rather dubious. They were either described and summarily lumped by most sources after that, or they were described and never recognized again, as the case like with Penstemon lepanthus by Pennell. So for this project, we looked at just at the plants with an index of A, B, or C, just so we had more confirmation and more confidence that these were worth recognition. Because as you can imagine, no 2 taxonomic sources agree. So if we just look at category A, B, and C, and one plant with no assessment, which we'll talk about explicitly later on, we had 65 presumed extinct taxa in the US and Canada. And I just want to show this visually because I'm always fascinated to show just how much agreement we have, and that most of the plants that are believed to be extinct that we recognized on this study were category A, just universally accepted, which is surprising because oftentimes you're looking at just a single specimen or few specimens as the hypothesis for a distinct species, but they're so different, they're recognized universally. More often than not, like very rarely, we have this B and C category, and then a pile of things in D or F which aren't recognized in the study. And all of these plants, B, C, and D category, or even F category, would be wonderful taxonomic studies because these may not have been fully evaluated, but they're already gone. So some of the results I'll share to kind of launch off from is that we had 51 species and 65 taxa that are believed to be extinct or globally historical in the United States and Canada. 42 taxa, and this was a real surprise, 42 taxa, or 64% are believed to be single site endemics, only known from one place on the globe. But we have to ask, were these really single site endemics or just under collected? And we'll talk a bit more about that challenge later on when we move into a prioritization effort to prevent extinction events. We'll point out that only 2 of the plants we recognize as extinct were listed by the IUCN as extinct. So there's a lot of work to be done on the international scale with IUCN. So if we look at the total flora of the US and Canada, about 15,882 species, it's about a 0.4% of the total flora is extinct, and you divide that by the years of publication, so it's about 3 plant taxa per decade are going extinct. Now we had a broad assortment of families. As you can imagine, the big families lost the most. Oftentimes, like Asteraceae with 8, Fabaceae with 7, Roses with 7, and Boraginaceae with 6. But then some big families are noticeably absent, like the grasses, the sedges, and the orchids. Now, I suspect the grasses and sedges are an artifact of under collections of these difficult groups, and the Orchidaceae there is because of over collection at the site of a single known species. And we had 53 genera, and this is a bit surprising. I don't talk much about the tree assessment that Murphy mentioned because she just covered that so well last month, but the hawthorns, Crataegus, those are trees and shrubs that are seeing disproportionate amounts of extinction. The genus Astragalus, well known in the Fabaceae. But then of great surprise to me was Cryptantha and Plagiobothris. Those 2 genera represent all 6 extinction events in the Boraginaceae, and they are evolutionarily sister groups within subtribe Eminiscinae. So it's quite possible there's something going on there, like a pathogen that hit evolutionarily close species in 2 different genera, because that is very striking to see 6 extinction events representing one of the largest families of extinction events being sister genera. Here's our map of plant extinctions in the United States. I'm going to point out really quick that there's a real heavy southwest slant to this map. This is most likely a combination of real, in fact, that there's this biodiverse hotspot kind of the southwestern United States, a lot of vascular plants, but it's also probably an artifact of European settlement. When settlers were landing in the east, they were developing open tree-less habitats, which can house narrow endemic species. And only when we worked our way west, we had the opportunity to inventory the flora before we destroyed places outright. So it's quite possible we lost many more species in the eastern United States before they were ever described to science. These would be called dark extinctions, and identifying dark extinctions is very difficult. Pointing out really quick, not much in the upper Midwest of the United States. We have surprisingly nothing in that panhandle biodiversity hotspot in Florida or places like the Lake Wales Sand Ridge. We have 1 extinction event in Canada, which is right here next to the line. This is Polygonatum biflorum variety Mullerium, which is a ITU of C, so it's a debated entity. But I'll also point out we have 2 broad-ranging species only. We had Micranthum and Micranthomoides, which started the story from New York to Virginia, and then Crataegus fecunda, which is located along major river systems in the eastern US. So the biggest losers, California, as you'd expect, with their vast flora, had 19 taxa believed to be extinct. And then Texas with 9 extinction events. We're hoping that some of those are only extirpated because they are known so close to the Mexican line, but no record, at least for all of those plants, is known from Mexico. So these are true global extinctions. Third place was a tie between New Mexico and Florida. And I like to talk about New Mexico really quickly because it highlights the challenges of working with such extremely rare herbarium material to find extinct plants. So for instance, the lost sunflower, Helianthus praeterminus, which has an ITU of A, everyone agrees it is different, is unmappable because the specimen says New Mexico. But from what I can gather, the search party must have been in Arizona when this collection was made. So sometimes we can't even nail down the state for which an organism was thought to occur. And then here in Florida, a lot of things down in very much South Florida where you'd almost expect, but again, other hotspots lacking extinction events. Now I'm going to jump to New England, which is a bit of a surprising tale for extinction in that there are 5 extinct plants in New England. Very unlikely that Florida only has 4 and New England has 5, which tells you something about some of the bias that exists in our understanding, our imperfect understanding of the extinction record. But I'm going to talk briefly about 2 plants in New England to tell unfortunate stories. And I'll come back to one of these later on because there's a real important project underway now. So I'm going to start with the slender, smooth crabgrass, Digitaria filiformis laeviglumis. This is a native, endemic, and rare, now extinct crabgrass that was found from a single site in Manchester, New Hampshire, on these thinly soiled granitic domes outside of the town of Manchester. It was described by Fernald in 1920, so a relatively recently described plant. It has an ITU of B. Almost everyone recognizes this as a distinct species or variety. Last collected in 1931, but what I want to point out here is that it is often very hard to know the reason a species goes extinct. And this Digitaria probably went extinct from a number of factors, but one of those factors is that Fernald himself, the person who described this plant, collected 31 herbarium sheets of this plant. And you'll see most herbarium specimens have multiple sheets, multiple rooting individuals. There are 38 known collections of this plant in the collection history that we're starting to compile from the databasing of herbarium material. And that is a completely unsustainable level of collection for a plant that must have been exceedingly rare from one place in New Hampshire. And unfortunately, that's not a one-off. Here on Cape Cod in Massachusetts, we have Juncus purvidus, the blunt-flowered rush. This is a single-site endemic to marshes outside of Hyannis on the Cape. It was last collected in 1927. It has an ITU of B. This is the plant that is sometimes considered a waif from Europe, but the Europeans have done their own analysis and say, no, this is very different from our plant, as it's sometimes included within the name Juncus subnodulosus. But this plant, again, Fernald collected 56 herbarium specimens of this plant. All of them have rooting material, and you'll see this was a clonal, large patch-forming plant, and there is no way 56 herbarium collections would be viewed today as acceptable. It's possible Fernald just assumed more was in the landscape, but I highly suspect his collection efforts here actually helped push this plant to extinction. So overcollection is a serious threat and something of concern when it comes to our rarest plants to pushing them towards extinction. Now, I mentioned earlier this idea of dark extinctions, which are extinctions that happened in the darkness, kind of unknown in the background. And since 1994, 4 species have been described or recognized as distinct from old herbarium specimens, and they're already extinct species. So top to bottom, Coriospermum pallidum is a species of the Pacific Northwest that was last collected in 1931, described new to science from herbarium material in 1995. Govinia floridana is an orchid from Florida, last collected 64, described only in 2000. I worked on Marshallia grandiflora, which I'll talk about in a second. So that was last collected in 1919, described or recognized as new in 2020. And then Potentilla uniginosa, which is a Californian plant that I believe Barbara Erder worked on, was last collected in 1974, but described in 2010. And I expect more dark extinctions to be recognized from the herbarium record as we have people looking at specimens that are geographic outliers. And that's how I discovered the situation with Marshallia grandiflora. So before this project started, Marshallia grandiflora, pictured here, was considered an extant and globally rare species of Pennsylvania south in a bimodal distribution to central Tennessee. And I knew this plant from working in the field in Pennsylvania because it's a G3. There aren't many G3 plants in Pennsylvania. It's proposed for federal listing or a candidate, and it's gorgeous. So I knew this plant as a botanist. So when I moved to North Carolina and studied their rare plant list, because after my career in Maryland, I took a job with the North Carolina Natural Heritage Program, I pulled up the herbarium specimens of Marshallia, or what they were calling Marshallia grandiflora, and I was completely blown away at what I saw. The plants on the left are what I was used to seeing in the high quality river scour of large river systems in the mid-Atlantic. And the plants on the right were these rather giant, robust plants of slow water systems or dry fields. The habitats are very strange, attributed to the species. So I reached out to Derek Poindexter and Alan Weekly and asked if they had done any work on this plant, which they had not. So we did our own study. I'll talk about it briefly. The cypsali, the fruiting bodies, are markedly distinct between the North Carolina plants called grandiflora on A, and then B, the plants that are called grandiflora from outside of North Carolina. And then we compared that to another very rare and apparently sister plant called Marshallia legrandii, which is there on the far right. We did some simple statistics, and the North Carolina plants come out as easily morphologically distinct from the rest of what is called grandiflora, and then legrandii here on the right. And biogeographically, they make total sense. You have what was called grandiflora here throughout the Appalachians in the Blue Ridge Physiographic Province. Oh, this is the Blue Ridge, I'm sorry. So this is the Appalachian Plateau here. And then you have the Blue Ridge Physiographic Province here where this now extinct Marshallia is found, and then the sister plant called Marshallia legrandii. But this gets confusing because though we've called these plants throughout the Appalachian or Allegheny Plateau Marshallia grandiflora, the type hails from Western North Carolina. So the name Marshallia grandiflora now applies to our extinct Western North Carolina plant that has not been seen in over 100 years before it was described. We had to rename what was called grandiflora, where it's extant. We called that Marshallia pulchra, the beautiful barbarous buttons. And here's Marshallia legrandii, which is a diabase glade species known from just 2 places in the world in the Piedmont of North Carolina. So Marshallia can have very rare species that are widely disjunct. One of the real surprises for me in working on the extinct plants was learning about extinct in the wild plants. Like, these are our black rhinos, and people are not appreciating what it's like to have extinct plants known only from the garden. And in our work, we work closely with Botanical Gardens Conservation International and some other partners to mine their databases against the extinct plant list to see what's been captured already by conservation gardens. And there are 7 possible plants that are believed to be extinct in the wild. 2 of these have question marks because the gardens holding the material have not replied even still with verification that these plants are identified correctly. So we're just putting them on the list as a question mark because we're not sure if they're truly extinct in the wild or if they're extinct proper. But one of the plants that really captured my imagination was Arctostaphylos franciscana, which is something we're going to have to think about as a community moving forward in the future, because this plant was thought to be extinct and then rediscovered by one of my co-authors, Dan, I'm going to mispronounce your name, so excuse me, Dan Klausenkamp. He rediscovered this along Doylestown Avenue in California, but it was part of a shovel-ready project in 2007 or 2008. There was no stopping this project, even a rediscovery of an extinct plant, which kind of shows something policy-wise we should think about, and that we should have policy where an extinct plant, when it's rediscovered, is automatically listed or proposed under some protection. But that's not the case, and that's fine. That's not why I'm here. But the community rallied and did what we could, and we dug up and moved this plant to a nearby area where it's doing well. But the definition of extinct in the wild is no naturally occurring populations. So now we have 5 extinct in the wild plants. That was from our study in 2020. But I'm saddened to report that we found yet another extinct in the wild plant very recently. This is Stephanomeria malheurensis. It's known from a single site in Oregon. It is listed under the Endangered Species Act as endangered, and a lot of effort's been put into preventing the extinction of this species, all of which have unfortunately failed. Between 2007 and 2011, over 4,500 seedlings are planted and watered for 8 weeks, in which all failed. 46,000 seeds were directly sowed at the site, and they have failed. 150,000 seeds are produced, and I believe in seed banks now, but all that action is put on hold because of the continued failure of the species at the site. So because there are no naturally occurring populations, this is just known in seed banks or gardens, this is now our 6th extinct in the wild plant. Which leads me to one of our side projects that's been kind of a growing informal collaboration, which I call Extinction Gardens. I'm working with Ray at the U.S. Botanic Garden, Mount Cuba Center. I've worked with other organizations like the Morton Arboretum, Portland State, and Santa Barbara Botanic Gardens to gather all of these extinct plants for a display. And we're still trying to figure out the details here, but I desperately want to put the public in touch with these extinct plants so they can see them and touch them and learn from them. We need to tell the stories of these extinct plants to show that plant extinction is real and you can interact with it. So right now we have 4 of the 6 extinct plants gathered at the Mount Cuba Center. The Stephanomeria is coming soon, and then we just have Prunus meridum, a variety Gravesii to get from New England, but there are a number of locations. This is the Arctostaphylos franciscana, which Santa Barbara Botanical Gardens mailed us for the exhibit. And this is, I just want to give a shout out to Ron Lance, a noted Crataegus expert who I've leaned on for years, who has been propagating some of these extinct Crataegus. Actually, the Morton Arboretum mailed Ron some cuttings of Crataegus fecunda after they found they had among the last plants in the world. He's been able to propagate numerous other individuals of this plant. Now, the cause of extinction is really almost impossible to determine unless the plant was known from a single site and that site's been destroyed. All other times, you're kind of dabbling into speculation about why a species went extinct. But it's pretty clear to see that when you have such a high percentage of single site endemics that are going extinct, that all your eggs are in one basket, and if a site's destroyed, the species is extinct. Even something as famous as Franklinia altamaha, we don't really know why it went extinct. There's speculation it was pathogen, fire alteration, overcollection. We just don't know. So it's even hard for single site endemic plants to discernibly know why a species went extinct, let alone these more widespread species like the Crataegus or the micranthemum that started it all for me, because the micranthemum was probably a death by 1,000 cuts. You have saltwater rise, sea level rise, ripraps of shorelines, ditching, invasive species. The number of things impacting that species are large, vast, and gross. But I feel like part of this is a call to action, because I'm not necessarily an academic scientist. I want to see on the ground conservation action. So I wanted to take this exercise to prevent extinction events, because preventing extinction is the lowest bar we can set for success. We need to actually do much more than just prevent extinction, but sometimes we're on the cusp and the precipice of extinction, so we have to work on those organisms because we're not succeeding. And that led to this project to identify and prioritize single-site endemics for ex situ conservation action. Now I'm going to use Sarracenia aureophila here is a wonderful example of why we need to prioritize. And this is not meant to be throwing stones at all. I'm just highlighting inequities and things that we need to work on. So if you look at the Botanical Gardens Conservation International database of collections housed in their gardens, you'll see, I hope you guys aren't seeing that taskbar at the bottom like I am. I don't know how to get rid of that. Let me try that and that. Well, maybe you can see it, I don't know. So you should see Sarracenia aureophila at the bottom of that screen. And you'll see that there are 44 ex situ global collections for that plant worldwide. That's a G1 pitcher plant. There are undoubtedly more ex situ collections for that plant off-site worldwide than exist on the ground. That's great. However, something like this Pachyra serpenticola, which is known from a single site in western North Carolina, the Buck Creek Serpentine Barrens, is known from no collections. So we have to ask ourselves, are we prioritizing the way we should be to prevent plant extinction events, especially when we know single-site endemics are disproportionately going extinct? So that's led to this collaboration with Ray at the U.S. Botanic Garden to identify single-site endemic plants. So when we started this, and I alluded to this earlier, sometimes it's very difficult to know if a plant was truly a single-site endemic. We have an idea in our head of what a single-site endemic is. It's a plant that's found at one place. It will probably only ever be found at one place. It evolved at this very special place. Like I'm thinking the Katona Dolomite Glade endemics, the Buck Creek endemics. There are many single-site endemic plants at sites like that. But there are other plants that were once more widespread, and they have declined to a single site. Now that plant, illustrated here with the two X's and the decline to one spot, would not be endemic to a single site, but it is now a plant of one known occurrence. So there's a subtle difference here between single site endemics and plants that are known from just one known occurrence, even though single site endemics are plants of one known occurrence, or OCOs. This is an umbrella term that captures single site endemics and plants of one known occurrence, because these are the plants we should be prioritizing for conservation action in situ and ex situ. So I'll show a couple pictures and how this works. So this is Arenaria livermorensis. I got to meet this beautiful little creature out in West Texas 2 years ago now. This is a global single-site endemic, the Livermore Mountain. It's known from just this spot in the world. It will probably forever be known from this small, isolated geographic area. But then we have plants like Ancystrocarpus kellii, which was known from two locations. But this other location here on the east side was last seen, I believe, in the 1930s. So this is declining to just one site in the world. So these are the kind of plants we're trying to identify for global conservation action. Those single site endemics and these plants have one known occurrence. And the data I'm going to present is preliminary for the Western US. The Eastern data is really good. That's why it's presented without asterisks. And I hope to expand this project into Canada someday, because once we have the method rounded out, and I don't have time to go into the methods today, but once we have the method rounded out, it should be relatively easy to generate a list of single site endemics or OCO plants from Canada for priority conservation action. So this is NatureServe's map of biodiversity importance, MOBI, which is a hotspot map of G1 through G3 plants, which we call at risk. So the hotspot map lines up where biodiversity would be expected, like in California, in the Panhandle, in the mountains, etc. If you overlay the single site endemics or the OCO plants on top of this map, you'll see they're where you expect for the most part. So there's 29 in California, 11 in Utah and Texas, respectively, and then a real smattering of smaller numbers across the United States with some surprises. Like, who would have thought Vermont or New Jersey had a global single site endemic plant species? And those are truly single site endemic plants. They're not known to have been found anywhere else and declined to a one known occurrence. Those are single site endemics. So you have to ask yourself, if this is what we need to be doing, maybe we're already doing it, but the data right now argues that we're not. So if we look at those one known occurrence plants and ask how we're doing, we're frankly not doing as well as we should be. So right now our data set has 89 plants on it, and we hope to round this data set out and finish this project this winter for dissemination and action. So there's 89 OCO plants known from the US, and only 9 of those are listed under the US Endangered Species Act right now. So there's a large number missing there if you feel like they should all be listed. And I understand just extreme geographic restriction does not qualify for federal listing, because Solidago plumosa here, the Yadkin River goldenrod, is known from a single site in North Carolina. It was petitioned and not listed because the landowner is interested in the conservation of that species. So there's more to it than just being extremely rare. So the CPC, the Center for Plant Conservation National Collection, has already captured 38 of these 89 in ex-situ collections, which is wonderful. And then BGCI has collected 55 of 89. So clearly there's a lot of work here to do to make sure the ex-situ component is capturing all of these species for extinction prevention. And frankly, we should check all the identifications housed at CPC and the BGCI plants for these to make sure we are actually capturing the organisms we believe we're capturing. But after this is done, the intent is to disseminate this data for priority on the ground and off-site conservation action. So I'm going to swing back around to some of the taxonomic difficulties in projects that we've broken out of the extinction work. Just to refresh your memory, this is that Digitaria filiformis laeviglumis from New Hampshire. One of the things we did in the paper, we put an asterisk next to this because it was reported as extant, which is the opposite of extant, from Mexico and Venezuela. And frankly, that blows my mind to think that the same plant known from a single site in New Hampshire would be found in Mexico and then again in Venezuela. I had my doubts, so we built a collaborative project with Bill Nichols of the New Hampshire Natural Heritage Program, Joseph Whipf, who did the Flora of North America treatment for Digitaria, Aaron Seigel at UNH, Jorge Ken, who was the person who reported the plant from Mexico, and then Craig Barrett's lab with some undergraduates to do a molecular study on the filiformis complex and to reevaluate this plant as, is it really extinct or not? But to get to the answer about what Digitaria filiformis laeviglumis was, we had to do a big molecular study and a morphologic study. So I'm going to present the preliminary results of the molecular study because making sure these organisms are worth recognition is important because that impacts where we put on the ground dollars, what we spend our time looking for rediscovery, and what we protect on the ground. So the filiformis complex consists of 4 varieties, Dolichiophila, filiformis, laeviglumis, and villosa. And one of the questions we wanted to answer is, are all those varieties real? Are they worth recognizing? Where are they found? And then also, so what are the plants in Mexico? Are they really the same as the ones in New Hampshire? We need to do some field work in New Hampshire. Is that plant really GH or is it truly extinct? So we had to work broadly and collaboratively to answer that question. So here's a picture from the New Hampshire site. There's that kind of granitic rock outcropping. And unfortunately, there is no Digitaria laeviglumis there. Numerous surveys have been done now, so we're going to move that plant to extinct. But unfortunately, 2 non-native alien species of Digitaria are found on site now. So it's quite possible we have the overcollection effort impact made by Fernald, where he collected 38 specimens, 31 specimens, including roots. And then you had the secondary invasion of these non-native Digitaria at this site, which has now really caused truly the death knell for that species. So the molecular results are really fascinating. Laeviglumis is an extinct plant, so we had to take like herbarium material for that analysis. But you probably might not be able to read this tree on the right really well, but I'll just point out that the nuclear ITS and the plastid DNA, their results match, and they're fascinating because Laeviglumis forms its own grouping, sister to the South Florida endemic Digitaria, and those two form sister to the Mexican Laeviglumis. So it's very interesting to see that result. We were surprised by that. And another thing we were surprised by is that Digitaria isn't tracked in the state of Florida, but it's only known from the pine rocklands. So this is probably a globally rare plant masquerading kind of under the radar as something that is not tracked right now. So a lot of conservation impact in asking basic questions about what species exist. So Joseph, the Flora of North America treatment author for Digitaria, is going to elevate all of these varieties to species. We're going to have Digitaria Digitaria laeviglumis, which will be the extinct plant in New Hampshire. He'll rename whatever he decides to plant in Mexico, because that is not the same evolutionary unit that we have in New Hampshire. We're going to move Digitaria laeviglumis to X, truly extinct, not potentially extinct. But then we do have to ask, what is up with this plant in Venezuela? The truth is, we couldn't get material for the study, but morphologically, there is no way that is the same plant of either the thing in Mexico or the plants in Florida or New Hampshire. So still more work to be done, but all really important conservation impact work. But we've had a lot of depressing information and stories today. So I'm going to leave us with some uplifting, fun projects that hover on the precipice of understanding species and extinctions. And this is a big collaboration. It's really been made possible by Ray at U.S. Botanical Gardens and Murphy Westwood at the Morton Arboretum, where they pulled numerous experts from across the United States to do multiple trips to Southwest Texas to look at the red oaks. I'm not going to mention everyone by name, you can read their names, but Adam Black at Bartlett Tree Services and Michael Easton of the San Antonio Botanical Gardens are just dreamy botanists to work with. They really know the flora down there. And this work on understanding species complexes has resulted in a few different field surveys. And I put this silly photo here, this kind of Botany Boys glamour shot, because those plant presses are completely full. We made a lot of specimens of various red oaks in Southwest Texas, complete with molecular vouchers, so we can figure out what the entities are that are worth recognition. Because when you think about conservation, what is it, where is it, how is it doing? You have to have good boundaries to identify the organisms in the field so we know where to put our conservation dollars. So we did 2 collection trips last year that included Dr. Andrew Hipp, who's leading the molecular component of the work in Chicago. And part of this was truly a hunt for Quercus tardifolia, which is what's thought to be an extinct species at the time. And here's kind of what the challenge we have in Southwest Texas. And I put the G ranks under each illustration. These are taken from the Flora North America project. So working left to right, we have entities like Quercus filiformis, which is G1, so among the rarest plants in the world, endemic to the Big Bend area of Texas. The next to that, morphologically similar, you have Quercus emorii, which is G5, abundantly demonstrably common. Then you have Quercus robusta, which starts to blend the lines and is also a G1, thought to be endemic to the Big Bend area of Texas. Then you have the GH, globally extinct Quercus tardifolia, which was really the focus for this work for me. And then the G5 Quercus gravesii, which is also abundant and common. So there's a lot of questions when you're in Southwest Texas. What are these trees around me and what names apply to them? But I was most interested in this plant, Quercus tardifolia, because it was one of our extinct plants on that 2020 study. It is more often than not recognized as distinct. It has an ITU of B. And this is the last known collection collected by Dr. Mike Powell from Big Bend. And what I want to point out to you is that among all the oaks in Southwest Texas, this has a wide leaf, very thick coriaceous leaf, and that kind of alba tomentose indumentum on the bottom of the leaf is really striking. None of those other oaks in that slide of illustrations I show you has that kind of arachnoid pubescence. So as part of this study, we wanted to answer, what was this plant? Because sometimes Quercus tardifolia is considered a hybrid of unknown parentage. So we wanted to get an answer to this. So we took some molecular samples, some cuttings from this material, as well as from the type. And that's really important to sample the type in this case, because it's only been known from a few trees in one specific location at Boot Canyon, last collected in 1933. Sorry, the slide's cut off there. But thank you, the California Academy of Sciences, for letting us sample that material. This is the holotype. We did not sample the holotype. We sampled an isotype. So to really survey for this plant was a multi-day arduous effort, as you can imagine. If you know anything about Big Bend, it's about as big as Delaware, where I'm from. So the idea of having a park as big as a state is really flabbergasting. But we realized in our previous field work that we needed to spend days and days in this canyon if we were going to rediscover the species, because it's not easy to get into. It's a multi-hour hike each direction to get in. And as Michael Eason famously said, we're only 45 minutes from the parking lot, and it took us over 2 hours to get back. So it's hard hiking. It's hard to get into. And if you want to survey this deep canyon, you need to stay put for a while. Now, Boot Canyon's called Boot Canyon because of this kind of geologic spire that looks like an upside down cowboy boot a little bit if you squint and nod. But to pull this off was a real effort. We had these mules that carried our camping supplies to our site. We had a camp manager named Kelsey. He really took a lot of burden off of us from an exploratory standpoint. The idea of hiking 10 plus miles a day in really arduous, rough terrain like this, and having to go back to camp and get water and cook food and prepare for the next day is a lot. So thankfully, a camp manager could help. But here's us kind of scampering down one of these bouldery canyons. This is not light field work. But I'll never forget the day. If you haven't seen the news story, we did rediscover this plant, which is super exciting. And this guy here to the right of the tree, that's Michael Eason. Michael is one of the best botanists working in Texas. He knows the flora. And we were kind of marching through this forest like a search party. And Michael, I heard him say, "Wes, you need to get over here." And the excitement in his voice still kind of gives me goosebumps because, you know, Michael knows the flora of Texas. I'm not going to teach that guy a new plant. So I knew he found something really good. And I walked over and was immediately blown away by this tree because he'd probably seen thousands of trees at this point throughout our multi-day trip in Big Bend. But this tree was clearly different. Even from the distance, you could clear, the leaves are so thick, the light didn't shine through them. So here it is. Here's a picture of it. Densely pubescent backside of the leaves, thick coriaceous leaves. And you see we're all smiling and happy. But the truth is, this is kind of sad. Yes, we rediscovered it, but at the time we had one tree. We have one tree. And it's not a healthy tree. Adam Black, that big tall fellow on the left with the 10-gallon hat, he kind of pointed out that this tree is fungally infected, that it's stressed out. You can see all the branching up and down the tree trunk. It had been burned over previously, probably the last year. So this tree is stressed, and according to Adam, quote, not long for this world. So we have to do some priority action now that we've rediscovered this global single site endemic. We need to do off-site conservation, that ex situ conservation. Let's get some cuttings, let's do some grafting, let's get a number of plants together, because this habitat where this tree is found now is not ideal for it. The habitat's changed over time, and this tree is kind of like locked in a sky island, and having additional plants to put somewhere else that we can mutually agree upon, probably at Big Bend, is probably the best path forward to preventing this species extinction. But there's a lot of work to be done as I round out this talk. When thinking about preventing extinction events, that global single site endemic project, that OCO plant project, to me is the most meaningful thing we can do at this time. Now, there's going to be a lot of plants that we vet that aren't single site endemics or aren't plants of one known occurrence. And we're going to present all of those as well as these OCO plants, because they're going to be among the rarest plants in the world, even though they don't meet our very strict definition of single site endemic. And this is where at NatureServe, I really want to unite the ex situ and in situ conservation partners. My entire career has been in situ conservation, on the ground, searching, exploring, protecting, land management. But we need to work more collaboratively with those conservation gardens to make sure we get the seeds we need to conserve the plants growing offsite, and make sure we know how to grow these plants from seed when we have the seed. Make sure things are viable. We just need to work together for the greatest conservation impact possible. And as part of this OCO project, we want to identify the on-site condition, like is it on a place that's being managed for the species? Is the site protected or not going to be converted to like a development or something? Are the seed bank collections correct? And then we want to publish and disseminate that data, because this is the real actionable component where conservation is meeting extinction. So in conclusion, there's just so much left to do. As I mentioned, some of these summary stats, I don't need to read them all again, but there are so many taxonomic questions that exist about some of these plants that if you're a researcher out there and you're interested in any of these groups, I'm happy to share the data I have. If you have a student interested in investigating some of these potential dark extinctions that are now lost to synonymy, that would be really wonderful. But one of the other take homes I just want to say really quick is that like the stakes are just really high here. We're talking about immediate actions to prevent extinction. Things are slipping through the cracks. We just need to work more collaboratively to make that happen. And this project probably started in 2014 or 2015. So there's a ton of people that I need to thank over the years, and the new partners who've become very important in this research. It's like Rusty Russell and Robin Everly at the Smithsonian. For them to allow me to set up camp and do the research was really, really valuable. And then my previous careers in the Maryland Natural Heritage Program and the North Carolina Natural Heritage Program really directly resulted in my understanding of the conservation of these plants. Gary Moore really stepped up with USDA plants and got that paper provided to be open access, so it's just more available to everybody and not paywalled. Ron Lance has been the best in working through that very difficult group of plants called Crataegus, the hawthorns. He wrote a book called Haws. I greatly appreciate it. But I'll point out the hawthorns are probably the most misunderstood genus in eastern North America and Canada. No two experts agree on what species exist. There's a long list of names in synonymy or that have been lost over time that could represent very narrow and now extinct plants. Abby Meyer and Nick were really great at BGCI for helping me vet data in their database. Matt Lobdell at the Morton Arboretum helped extensively to make sure that the plants, the Crataegus fecunda at Morton was accurately identified. I want to call out Marianne Renault really quickly because she's done a couple of really wonderful press-related items. If you Google about extinct plants or Quercus tardifolia, she's really helped share that news, as has Matt Candidis. And then lastly, I know Peggy's on the call. I don't know if David's on the call. They're just general support for all things NatureServe botany directly impacts our understanding. And that goes for those NatureServe member programs too. This cannot be done without the collective efforts of everybody here. So I'll leave you with a beautiful single-site endemic from Georgia, Trillium georgianum. There's my email, really easy, wes@natureserve.org. I am more than open to criticisms or critiques or other ideas. So please reach out if you have any thoughts or comment. Thanks for having me.